Hello, Dr. J here with another exciting video lecture module. This one is about meiosis and spermatogenesis. Remember, meiosis is very similar to mitosis. We start with a cell that divides, copying its DNA and splitting the organelles and fluid into two different cells. What makes meiosis different is that there is a second cellular division. So we start with one cell and we end with 4. Another significant difference is that instead of trying to maintain identical DNA patterns like we do in mitosis, intentional hybridization of the DNA occurs. In other words, there's a scrambling of the DNA because we want each sperm and egg to be genetically unique. Now, when I'm growing a bone cell or a skin cell, I want them to be my bone cells and skin cells. I want them to be just like the bone cells and skin cells that I've always had. But when we're making babies, we got to scramble the DNA because we don't want our children to be exact clones of ourselves. Not only would that be boring and annoying, but diversity is a way that life spreads the risk, right? The more genetically diverse a population is, the more likely it is that somebody's going to survive if something bad happens. Maybe there's a killer pandemic that comes through. We don't want everyone to be exactly the same, or we're all going to get sick and die. Each of us is a little bit unique in terms of our immunology. Likewise, if there's a change in the environment, we want our children to be adaptable, and we want some of them to be a little bit different so that somebody can take advantage of a new situation. So anyway, sexual reproduction is really important biologically because it is one way that evolution helps to sculpt the population to be best suited to its environment. Okay, so that's all the background stuff. Here's the detail you need to know. At the beginning of meiosis, these chromosomes find their partners, their homologous partners. In other words, remember, this is the first chromosome that I got from Dad, and this is the first chromosome that I got from Mom. This is the second chromosome I got from Dad, and the second one from Mom. Third, fourth, remember there's 23 pairs, and so the illustrators have done us a great favor by not showing all of them. They've just showed us 2 pairs. That's going to be the simplest way of trying to get this hybridization concept across. Okay, so imagine if I had a party, and everybody came, and everybody brought their boyfriends, girlfriends, wives, their husbands. When the slow music starts, are you going to have a hard time finding your partner across the crowded dance floor? No, you're not going to start dancing with my wife. I'm going to not start dancing with your girlfriend. We're going to find each other. And so in this same way, the first paternal chromosome does not match up with the second, but only with the first maternal chromosome. So in this first stage of meiosis, prophase 1, notice that we have to denote the first, not the second cellular division. So we use that Roman numeral. In prophase 1, crossing over occurs, whereby a small portion of the maternal first chromosome latches on to the paternal first, and vice versa. So at the end of that hybridization, we got a blue chromosome with a red leg, and a red chromosome with a blue leg, right? Just representing the fact that they've swapped some genes. Then, in metaphase, we line everybody up in the middle, and in anaphase, we separate them. So it really becomes significant which genes from Dad ended up on this side of the line, and which genes from Mom ended up on that side of the line. It's technically called random assortment or independent alignment. Either of those 2 terms is fine. And that happens in metaphase 1. So crossing over and random assortment are two ways that this process, meiosis, guarantees that these sperm or egg will be genetically unique. So a man and a woman could have, well, theoretically, hundreds of children, but because every egg she ever ovulates is different, and because every sperm he makes is different, every single one of their 100 children would be genetically unique. Oh, unless they were twins, and we'll talk about that in a future video. So what do you really need to know about this? Well, actually, most of the language that's on the screen. You should be prepared to put these stages in the proper order. And like we did in our last chapter, you should be prepared to tell me significant events of each of these stages. So here's another example of a slide you could print and cut apart, put it back together as a puzzle, as a way of making it active so that you know which stage comes first and second, so on and so forth. Just as a very quick aside, it is during this process that things sometimes go wrong, producing an egg with too many chromosomes or too few. Less commonly, but also possible, the sperm could be problematic, right, in terms of the number of chromosomes. So in that case, if we had a sperm or egg that had too much genetic material, when they combine, we end up with something called a trisomy. One good example of a trisomy is trisomy 21, more commonly known as Down syndrome. If we look at this graphic, we can see characteristic facial features, right? A lot of kids with Down syndrome just sort of have a look about them. And the reason is there are key characteristic chromosomal abnormalities. And all kids with Down syndrome have this same chromosomal variant, and so they have a commonality in their heart defects, in the way their brains develop, in the way their hands develop. So we can see it right here. Down syndrome is trisomy 21 because during the development of the egg and meiosis, the egg had two chromosomes from 21, and during fertilization, the sperm brought the third one. So whereas it's typical that there should be only a pair, at the 21st position, we see three. Now, this is not the only trisomy. It's just that most of the other trisomies are fatal, and so we don't see these individuals in the community. Some trisomy individuals with trisomy 13, for example, can survive, but oftentimes they have such profound birth defects, cognitive disabilities, that we don't really see them out in the community like we do with kids with Down syndrome, who are usually leading normal and happy lives, able to communicate and participate in the community. Okay, anyway, just a really quick aside there on meiosis. Now, what you want to do is watch the videos embedded within this PowerPoint, and as quickly as you can, try to apply what we just learned about meiosis to the specific process of making sperm. It's called spermatogenesis. So let's see if we can put these two stories together. The little kid hit puberty, 14-year-old burgeoning young man is now capable of producing sperm. It's really the presence of luteinizing hormone and the androgen binding protein and testosterone all sort of coming together to turn on this type A spermatogonium. This is a fine graphic, provides a lot of good information. My one criticism is if I were the illustrator, I would have put a little loop right here, reminding us that before this process starts, our type A spermatogonium has to undergo mitosis to clone itself, so that there is always one of these around. There's actually millions of them, but the point is, if you just transformed the type A into a type B and then made sperm, you could only do it one time with that cell. And that's not how it actually works. You probably know healthy individuals are capable of producing sperm well into the 70s, 80s, 90s, maybe even up above 100. So the type A spermatogonium is sort of the wellspring, continually producing a lifetime supply of the type B. So this is actually mitosis that happens first. Then the type B spermatogonium migrates toward the opening, the lumen of this seminiferous tubule, and at which point we call it a primary spermatocyte. The primary spermatocyte undergoes the first cellular division, meiosis I, to become secondary spermatocytes, which undergo meiosis II to become these spermatids. These now have unique genetic makeups, right? Genetic fingerprints, but they don't have tails yet. They don't have their motors yet. So spermatids become sperm through the very last process called spermiogenesis. Now let's pause there just for a second and consider how many words we just learned that have sperm or spermato in their name, and you got to know all of them and know which ones are processes and which ones are cells. You have to know which cell undergoes which process to become which other type of cell, all by our first lecture exam in short order. So, for example, you should be prepared to answer this type of question. Which cell undergoes meiosis II to become spermatids, secondary spermatocytes? Which cell undergoes mitosis to become a type B spermatogonia, so on and so forth? We'll have some activities in class to reinforce these ideas, but you want to make sure that you are comfortable working through these slides and the following videos before next week. The very last thing I just want to introduce is that sperm have a very special sac that contains enzymes. It's called the acrosome. You can see it here. So during that last process, spermiogenesis, in addition to growing a tail, it grows a head containing both the nucleus and this special package, the acrosome. The sperm uses the acrosome to hack into the egg, a process that we'll take a look at in closer detail in an upcoming video.